Biomolecules are the all organic molecules which are present inside the living system or a living cell are called as biomolecules. We know that if you take any living organism, it is made up of various types of elements which may be either organic or inorganic. So all the organic molecules which are present inside a living cell are called as biomolecules. Why we are saying biomolecules is, here bio means living. That means the organic molecules which are present inside a living system or inside a living cell. Okay, here these biomolecules are made up of various types of elements like carbon, hydrogen and oxygen. Apart from this carbon, hydrogen and oxygen, they are also made up of the other elements like nitrogen. So these are the four major elements by which the biomolecules are made up of. So let's come to the other side of these systems, that is when you come to non-living systems, like what is a composition that is present in the non-living system of the non-living organisms, or let's take an example of earth crust. So earth crust is also made up of the same type of elements by which the living system or living cells are made up of. Then what is the difference between the non-living systems like earth crust and the living systems like living organisms? The only difference is, okay, the proportion of the carbon and hydrogen, that is the abundance of carbon and hydrogen in the living cells when compared to the non-living systems. But in other part of other part of this one, we can say that the elements which are present in the non-living systems are present in the living systems also. Then how can we know that which type of elements are present in the living systems? So that can be done by a process called as elemental analysis. Then by elemental analysis, we can know the various types of elements which are present inside a cell or inside a living system. And after knowing about the type of elements, we have to know about which type of compound is present. So then after knowing about the type of compound, what is the molecular formula, which type of elements are present in that molecule have to be known. All that is done by various types of process, that is called as chemical analysis, analytical analysis, and also by doing the ash test or ash analysis test. So how can we know that presence of various types of elements or compounds in a part of any organism is? So first we have to dry the part of the plant, so that when we dry the part of the plant, all the water present in the plant body will be left out. And after drying up, we have to burn it. After burning that, we get the ash, and when we analyze the ash or when we test the presence of various types of elements in the ash, we come to know that various types of elements and also compounds which are present in that plant body can be known. So this is what we say that analysis or the ash analysis, which gives the information about the presence of various types of elements and also the compounds which are present in the living cell or any living system. Then here all the macromolecules or the micromolecules, we can say that the molecules which are present or the organic molecules which are present inside a living organism are called as a biomolecules, and these biomolecules are majorly made up of carbon, hydrogen and oxygen. And here which type of organic molecules are present in a living system? A living system may be made up of various types of organic molecules, like it may be proteins, amino acids, polysaccharides, sugars, nucleic acids like lipids, fats, fatty acids, like various types of organic molecules are present. And all these organic molecules are coordinated somewhere with other type of organic molecules, such that the process or metabolic process is going on inside a plant body. Likewise, knowing about various types of organic molecules is studied under biomolecules. Now let us discuss about the questions, and when we are discussing the questions, let us know something about the properties, the characters and functions related to various types of biomolecules which are present inside a plant cell and also in the animal cells. Question number 1. The acid used for grinding the vegetables or piece of liver to analyze the organic compounds is options: 1. Tricarboxylic acid, 2. Acetic acid, 3. Trichloroacetic acid, 4. Malic acid. Okay, here grinding the vegetables or a piece of liver, this is a technique or the process which is used in the in the process of knowing about the type of organic compounds or organic chemicals which are present in the living cells. So for this process, what we do is we take a piece of vegetable or a piece of liver of any animal, and that have to be done with crushed with the help of pestle and mortar by using a chemical called as trichloroacetic acid, that is Cl3CCOOH. This is a trichloroacetic acid, which is a chemical used for analyzing or knowing about the presence of various types of organic compounds present in a plant cell or a part of vegetable or a part of liver that is in the animal cell. By using this trichloroacetic acid, a slurry is prepared, and that slurry is filtered, and we get two parts. One is called as the filtrate and the retentant, that is acid soluble fraction and the acid insoluble fraction. In the acid insoluble fraction, we can see the presence of that is both in the acid soluble and acid insoluble fractions. Whatever we are getting, we can know the presence of various types of organic compounds present in that living organism or in that part of that plant body or the animal body. So question number 1, correct answer is 3. Question number 2. All the carbon containing molecules present in the living tissues are commonly called. Options: 1. Heterocyclic molecules, 2. Heteropolymer molecules, 3. Biomolecules, 4. Dicyclic and heterocyclic molecules. Okay, let's come to the first option, heterocyclic molecules. Heterocyclic molecules means the molecules which are having cyclic nature, and those two cyclic nature are different, that is they contain at least 2 different types of cyclic compounds in that one. Then second one is heteropolymer molecule. So the word polymer means it's an giant molecule which is made up of repeated units, and each unit is differing from one another. Such a polymer is called as a heteropolymer molecule. Then third one is biomolecules. All the molecules which are present in a living system or in a living cell are called as biomolecules, and these molecules are nothing but organic in their nature, and they are mainly made up of carbon, hydrogen and oxygen. Then fourth one, dicyclic and heterocyclic molecules. Dicyclic means a compound which is made up of 2 cyclic structures, and those 2 cyclic structures are differing from each other. So we call dicyclic and heterocyclic compounds. Okay, when you come to the conclusion part, so we can say that all carbon containing components which are present inside a living cell are called as biomolecules. Biomolecules are nothing but the organic molecules which are mainly made up of carbon. Apart from that one, hydrogen, oxygen is also there, and this type of organic molecules are present inside a living cell. Question number 2, the correct answer is 3. Question number 3. The major percentage weight of element present in the human body among all other elements compared to earth crust is that of. Options: 1. Nitrogen, 2. Carbon, 3. Oxygen, 4. Calcium. Okay. Here when you are talking about the type of elements present in the human body, we know that the human body contains various types of elements, but the composition wise, if you take the percentage wise, it is differing from one another. Out of the given options, we can say that oxygen is the one which is of 65% by weight. That means if you say that all the elements which are present in a human body are of 100%, out of that 100%, oxygen itself is occupying about 65%. So that major percentage weight of the element present in the human body is of oxygen, which is of 65%. So question number 3, the correct answer is 3. Question number 4. The primary structure of protein is formed by. Options: 1. Phosphodiester bonds, 2. Peptide bonds, 3. Glycosidic bonds, 4. Hydrogen bonds. Okay, what is a protein? Protein is an polymer of amino acids, or we can say amino acids are the building blocks of protein. And here in a protein, many amino acids are there. And one more thing, that basically each protein is made up of 20 types of amino acids. 20 types of amino acids. And these 20 types of amino acids may be repeated many times. And one more thing, that it is not compulsory that all proteins should be made up of the 20 types of amino acids, but the maximum types of amino acids that are present in a protein are 20 types, and they may be repeated n number of times. N number of times is many number of times, and if similar type of amino acids are there in one protein, we call it as a homopolymer. If different types are there, we call it as a heteropolymer molecule. And how does the protein is forming into chain like structure means, it is by a special type of we can say strong bond, that is called as a peptide bond. And how does a peptide bond is formed means, for example, if you take some of the one part of the example here, that this is a basic molecular structure of a amino acid. Consider this is one amino acid and one more amino acid is there here. In each amino acid there are three functional groups. One is called as carboxyl group, amino group, and R represents the alkyl group. Okay. So likewise here, the first, if you consider this as first amino acid and the second amino acid. During the formation of bonding, what happens is the bond is always formed between the COOH of earlier amino acid to the NH2 group of the next amino acid. When the bond is formed, one water molecule is lost from this one, and the type of bond that is formed is called as peptide bond. In the same way, the second amino acid is binded to the third amino acid, in which the COOH group of second amino acid is binded to the NH2 group of the third amino acid. So likewise, a chain of amino acids is formed with the help of an peptide bond. And now the protein chain which is formed will be a straight one, without any helices or without any coilings. And protein chain which is not having any helices or coilings is called as a primary structure. And one more thing, that proteins not only form the primary structure, they also form secondary structure, tertiary structure, and the quaternary structure. So let us point out what is the difference between primary, secondary, and tertiary here itself. Primary means the protein chain without any helices is called as primary structure. Secondary means it forms a helical structure, that is a secondary. Tertiary means the coiling is overlapping one above the other. And quaternary means chains of proteins will be overlapping one above the other. So likewise, we can see the difference between the primary, secondary, tertiary, and the quaternary structures. And in the primary structure, the structure is stabilized by the formation of the peptide bonds. So question number 4, the correct answer is 2. Question number 5. The simplest and least carbon containing amino acid is options 1 glycine, 2 serine, 3 lysine, 4 alanine. Okay. The given options: 1 glycine. Glycine is a amino acid which is having 2 carbons in it, that is C2H5O2N. This is a molecular formula of glycine molecule. Second one is serine. Serine C3H7O3N. It contains 3 carbons in it. Okay, second one is serine. The molecular formula is C3H7O3N, which contains 3 carbons in it. The third one is lysine. C6H14O2N2. So this is a amino acid which contains 6 carbons in it. Then fourth one is alanine. C3H7O2N. It also contains 3 carbons as like in serine molecules. So here these are the 4 compounds that have been given in the 4 options, and these are the molecular formulas, that is glycine C2H5O2N, serine C3H7O3N, lysine C6H4H14O2N2, and alanine C3H7O2N. And out of these 4, glycine is the molecule which is having least number of carbons, that is 2 carbons are present, and the one which is having the least carbons and also the simplest molecular structure. So question number 5, the correct answer is 1.